Question 6 asks about the mutagen hydroxylamine. Hydroxylamine is a chemical mutagen. It adds a hydroxyl group to cytosine and converts the cytosine into a hydroxyl aminocytosine. Essentially, this means that the mutagen adds an OH group to the amino group of cytosine. This makes cytosine only able to pair with adenine rather than the usual guanine. It's important to note that this mutagen only acts on cytosine and no other base pair. The first part of this question asks how cytosine reacts with different stop codons. Here we can see all the possible stop codons and their DNA strands. The top is the template strand and the bottom is the coding strand, and this is the mRNA that would result from these DNA strands, and they are all stop codons. If we expose these DNA strands to hydroxylamine, it would react only with the cytosines. So in this first example, it would react with the cytosine in this DNA, and instead of base pairing with guanine, it would base pair with adenine. This would change the subsequent mRNA This mRNA would now be UAA. However, we can see that this is still a stop codon. While the hydroxylamine would react, it would not change the subsequent codon. This is the only codon that codes for stop, however, that is not affected by hydroxylamine, as there's no cytosine for it to react with. This is our final stop codon. The hydroxylamine would react with this cytosine, causing it to base pair with adenine. The mRNA would then be UAA. Again, this is still a stop codon. We can see that hydroxylamine does not change any of the stop codons. Even if it changes the mRNA, it still eventually codes for a stop codon. Part C asks, if your original stop codons in Part A were caused by single base mutations, could they be reversed to their original non-stop codons by hydroxylamine treatment? The answer to this question is no. Hydroxylamine only reacts with cytosine, so it would not affect any other base pairs in the stop codons. The only outcome of treating DNA that codes for a stop codon is to just create a different stop codon. In conclusion, the answer is no, you could not treat it with hydroxylamine treatment. Part D asks, to choose a stop codon whose sequence is unaffected by hydroxylamine treatment, and assume that this is the first of two stop codons in a gene. Describe a possible intergenic suppressor point mutation that would allow for a longer transcript of mRNA. So this question is asking for an example of an intergenic suppressor mutation. The definition of this is a mutation that occurs in a gene other than the one with the original mutation. So here I've written out that example of the mRNA. This would be the first stop codon, aka the first mutation, and this is the second stop codon that we're not going to look at. The point of an intergenic suppressor mutation is that it's happening on a different part of the DNA in a different gene. Here's our first gene, and here's our second gene where that suppressor mutation is going to take place. The point of that suppressor mutation is to create a longer mRNA strand and essentially silence that stop codon so that it doesn't actually stop transcription. The key to this is that this second gene needs to encode for tRNA. So in this is the normal mRNA, or this is the normal DNA before it is mutated. It is encoding for this normal tRNA. This has the anticodon AUA. This would eventually bind with UAU, which codes for tyrosine. However, we're going to put in a second base substitution mutation on this first base pair. This is then going to code for a mutated tRNA with an anticodon AUU instead. When this goes through transcription and translation, the AUU is going to bind to the UAA and make tyrosine instead of stopping the transcription. The whole point of that suppressor mutation is to silence the stop codon so that we can have a longer transcript of mRNA. The next part asks, based on this mutation, describe two types of DNA repair mechanisms that the cell might use to fix a mutation in the correct strand. So this is the mutation that we're going to be looking at. The two types of repair that can be used are mismatched pair and base excision repair. First, mismatched pair enzymes. For the mismatched pair enzymes cut out a section and fill it in with new nucleotides. The enzymes do this by using the original strand of DNA as a template. The enzymes need to be able to differentiate between the two strands. They do this by the presence of methyl groups in the old strand. The second way that we can repair this mutation in this DNA is by base excision repair. This happens by recognizing the defective bases and removing them by glycosylase. And the nucleotide is then removed by AP endonuclease, and DNA polymerase brings in the appropriate base pairs. DNA ligase then creates a phosphodiester bond and reseals the DNA strands. Finally, let's pose a what-if question. Here we can see a strand of DNA that codes for an operon. Let's say for this example, it's a lac operon in a bacterial DNA. What if we exposed this DNA to the hydroxylamine? The hydroxylamine would react with all the cytosines in this strand of DNA. For example, let's say that gene 2 has base pairs A, T, and C. If we applied hydroxylamine, it would react with this C, and eventually it would pair with an A instead of a G. When that RNA polymerase comes in and binds to the promoter to read the strand of DNA and transcribe these genes into mRNA and subsequent proteins, it's going to misread the genes and create the incorrect proteins. This is what would happen in a different situation if we exposed a lac operon to hydroxylamine.